Hello. So now we're going to talk about clustering. What is clustering? It's nothing else than grouping, than trying to find within a set of samples which of them are more similar to each other. How can I group them best? So basically, you have a set of points and you're trying to divide them into groups. It's not very complicated. There are many ways of performing clustering, and we are going to look into some of them today. We're going to look at some non-parametric clusterings. So they are not based on a model, they are quite flexible and they can be used with any kind of data. And we find both hierarchical and non-hierarchical methods. And we're also going to look at parametric methods, which are based on a statistical model that assumes that there is certain properties to the data that we're looking at. So first, let's talk about hierarchical clustering. Now, hierarchical clustering is characterized because there is a cluster hierarchy. And the hierarchical clusters are expressed in dendrograms, which you have probably seen because it's how phylogenetically analysis is done. Now, this hierarchy means that clusters are nested within each other. So you first have, you define this small cluster, and then you put this cluster into a bigger cluster, and this cluster into a bigger cluster, and so on. To obtain hierarchical clusters, or to obtain dendrograms, we need to use distance measures. So we start from our data, calculate some distances between our samples, and then based on those distances, we can create a hierarchical cluster. And there are different algorithms to generate those clusterings. The most common ones are UPGMA and neighbor joining algorithms. And importantly, by cutting the tree at a specific height, we can obtain the division of samples into clusters. So if we cut at this height, we obtain 2 clusters. If we cut at this height, we obtain 7 clusters. Quite simple. Let's talk about distances. So the most typical, classical, and common distance is Euclidean distance. Euclidean distance is based on the Pythagoras theorem, and it just says that given 2 points that are defined by a series of values in n dimensions, distance between these 2 points will be equal to the sum of the squared differences, the square root of the sum of the square differences of those values. So to put it into a bit more understandable example, we can imagine that we use 3 variables to define 2 plants: height, leaf length, and anther length. And we have for plant P and for plant Q the values of the different variables. Then we just take 1 minus the other squared plus 1 minus the other squared, and so on. We sum them, then we do the square root of this, and we obtain the distance. One thing to realize, however, is that if you look at these numbers right here, that height is having actually way higher impact on the final value of the distance than the other variables. And this is because height is expressed on numbers that are larger, on different scales. And this is important because it means that in the end, if we do this distance analysis among many individuals, the distances will not say how different individuals are across variables, but how different individuals are across height only. And that's not what we want. So instead, what we can use is scaled Euclidean distances. So this is not a different type of Euclidean distance, but it's a distance where we use instead scaled variables. The general way of scaling is to just take the observation minus the mean over all individuals and divide it by the standard deviation of that variable, and then you obtain the scaled sample. Now let's imagine that height has an average of 100 and standard deviation of 10, and leaf length an average of 10, and anther length an average of 1, and with the standard deviations. If we would apply this formula to the numbers that we have just seen, we would obtain instead these numbers. And if we calculate the distance now, we can see that the 3 variables are giving more or less similar contributions to the final distance. Don't worry about these formulas right now because you will not have to do any of them. They are already programmed inside R. but it's useful for you to know what is actually going on and why it's important to scale variables. So, to talk a little bit more about different types of distances, we've talked about Euclidean distances, and the Euclidean distance measures whether two numbers are the same. So the more different two numbers are, the larger the distance will be. But there are many other distances with different properties. Manhattan distance, for instance, is very similar to Euclidean. It has a little bit of difference. The Pearson correlation distance, which is just 1 minus the correlation between two variables, measures whether the features in these variables change together, rather than whether the number is the same. And they change together either positively, so together, or negatively, opposite to each other. Then we have Jacquard distance, which is used for presence-absence data, and it measures just how many things coincide or don't coincide between two data sets. And it was used a lot for electrophoresis band analysis. Then we have many different types of genetic distances that measure different things within the genetic information, and of course there's other kinds of distances you can find out there. Today you'll be using Euclidean distance and some other distances of your choice, as you will see. After hierarchical clustering, we have non-hierarchical clustering. And we're going to be looking today at two methods: principal component analysis and k-means clustering. Now, before we continue, I really need to say that principal component analysis is not a clustering method. It's related to them, but it is not a clustering method. However, it is a method that allows us to further understand the variables that we are using and the data that we have, so that's why we are going to look at it today. Let's talk about principal component analysis. Now, PCA is a bit complex, so I will try to give you a bit of an intuitive understanding of what it is, because we don't have today time to cover the maths of it. PCA is nothing else than the decomposition of a matrix of correlated vectors into a matrix of uncorrelated vectors. What does that mean? Imagine that we have a matrix with only two columns, one that represents height and the other weight of a series of individuals. What PCA will try to do is first try to find one principal component that is the linear combination of height and weight, and that can hold as much of the variance as it can, as much of the original variance as it can. So in the case of this example that we have seen, this linear combination is equivalent to a line, a regression line actually, through all those points. And now I'm showing you here a 2D representation, but this can be done with any number of dimensions, and that is the power of PCA. It finds one variable that can summarize as much as the total variance of the dataset in one single variable instead of spread across many variables. Now first it will look for the line that can contain the most variance, which in this case is this line, and then each point would be expressed as a point in this line, right? Here or here or here. And then once it has created this first principal component, it will try to do a second principal component to explain the remaining variance that is uncorrelated to this first one. And in geometrical terms, uncorrelated just means perpendicular. So it will create a second principal component that goes in this direction, and where each point is expressed as a point in this line. Now, if we would apply PCA to this set of points, what we would obtain is first principal component and second principal component. And as you can see, in this case, because we're looking at a 2D problem, we have just rotated the points. And this is actually what PCA does also on higher dimensional scales, but it's not so easy to visualize. So the message is that each principal component is a linear combination of the original variables. Now let's look at it in a more complex setting when we have more variables. This is the PCA, so where each point is one variety of the Brassica varieties from 2007 that were evaluated by students with phenotypes that they had designed by themselves. Now, what we can ask to a PCA is two things. First off is what amount of the original information is contained in each principal component, and we can answer that with the percentage of explained variance, which I have plotted here and here. So this means that across this axis, there is 26% of the original information that we had in the total matrix, and in this axis, we have 25% of the information. So in total, this plot represents over 50% of the original information that we had. Yeah, so this is one thing. And the second thing that we can ask is what variables contribute and how much to each principal component. And for that, we can use the loading vectors. And we can plot those loading vectors on top of the PCA plot, and then we obtain a PCA biplot. Now, loading vectors can be read by looking at the size of the vector and the direction of the vector. Loading vectors, they represent the projection of each variable onto the principal component plane. So that means that the directions of the vectors represent okay, I don't know exactly how to explain it, but you can interpret loading vectors very easily if you think of them as extra axes in the plane. So whatever is very high on the leaf hairiness axis, so it's very going to this direction, whatever point, it means that this, for instance, this pak choi is very high on leaf hairiness, whereas this Chinese cabbage is very low on leaf hairiness because it's on the other side of the axis. But the direction also tells you something about the correlation of vectors. Parallel vectors are very correlated. So for instance, wax and height and flowering and lobes are all very correlated. That means that any plant that has high height will also have high wax and high flowering and high lobes. And if two vectors are going in opposite directions, it means that they are negatively correlated. High leaf hairiness will mean low plant shape. Perpendicular vectors are uncorrelated. So, for instance, if you go higher on leaf hairiness, it will not you can go higher on leaf hairiness but not affect your lobes variable, if that makes sense. So that's how you interpret the PCA. With a combination of the points and the vectors, you can know more or less each point that you have, how much or how little it has from variables, and you can also see how the variables relate to each other. One important thing to note is that PCA is also sensible to scaling. I've been showing you now the PCA of unscaled data, but if we scale the data, then we obtain this kind of PCA. And what you can see is that suddenly the directions of the loading vectors have changed, and also the sizes. This actually happens because with the unscaled data, plant height, leaf hairiness, height, and stand petiole were having the most impact on the PCA and on the distribution of points in the principal component plane. Whereas with scale data, all variables start to have an effect on the PCA, more or less in a similar, with a similar dimension. And that's more what we would like. We would like all variables to impact PCA similarly, rather than some variables having way more effect. So with this, I think we finished PCA. You probably have some questions, but hopefully we can communicate through some online channel to solve those questions. The next technique that we're going to talk about is k-means clustering. And k-means clustering is just a way to answer the question, how can we divide our data in k groups? So the way that k-means works is the following. You have a set of points, and then you randomly assign points to k clusters. You calculate the centroids, the means of those clusters, which are these 3 points that you can see here in the middle, and then you reassign the points so that the cluster that they belong to, so they are closer to one How do you explain this? You reassign the points to minimize the point centroid distance. So that means that the points will become part of the cluster that they are closest to. No, that doesn't make sense. Then you reassign the points to minimize the point centroid distance. Finally, you recalculate the centroids and then you reassign points, and you repeat this process until everything is until the situation is stable. Now, one thing to note here is that we start from a random start, and therefore the end that we achieve might be different every time. So, because every time we might get a different result, we need to check which one is the best result out of all of the results that we have. And this is something that R does automatically, and we will see later how that works. So, in order to decide what is the best result, we need to be able to evaluate the results. And for that, I would like to go back to the definition that I gave at the beginning. In the end, clustering tries to find clusters in which the objects within the cluster are more similar to each other than those in other groups. So, how do we check that? We can use a measure called within sums of squares. You will probably see these formulas later in advanced statistics, so I'm not going to go into detail with them, but you just need to, well, I'm just going to give a brief explanation. So, the difference between an observation and the mean of its group, the sum of all of those differences within a group will be the within sums of squares. So that is just the distance of each point to the center of a cluster. Then the between sums of squares is the distance between the center of each cluster to the center of the total data points, which is just the... And finally, the total sum of squares is the distance between the points and the center of all of the points. So what we would want to get is clusters where all the points are very close to the center of the cluster and the clusters are very separate from each other. So what we want to get is a clustering where the within sum of squares is very low, but we have high between sums of squares. That is, most of the variability of the data is between the clusters and not within the clusters. So this formula we cannot only apply to decide which of our random results is best, but also to ask ourselves, okay, if I can put any k, so if I can divide my clusters in any group of clusters, any grouping, which k should I use? Which is the most adequate? And for that, we can use the within sum of squares, as I have been suggesting. So in this plot, what we can see is that as the number of clusters that I put, as the number of k that I give increases, the within sums of squares decreases. When I have one cluster, of course, all the variability is within the cluster because it's only one. And as you increase the number of clusters, the amount of within sum of squares decreases. But then you will realize it just continues decreasing forever. And of course, that makes sense. If you increase the number of clusters, then the distance of the points to the center of that cluster will diminish, and there will be a point where if in each cluster you have one single point, then there will be no distance between the cluster points and the center of those clusters. So it doesn't make sense to just increase the number of clusters forever until my within sum of squares is 0, because then it's not informative. So you need to ask yourself, okay, at what point is the decrease that I am having significant because I am separating better my observations, or am I just having a decrease because I am adding another cluster? And I find it a bit hard to see it in this kind of plots, and instead, so yeah, we need to ask ourselves how to, at what point are we still improving significantly? I find it more easy to see in the within sum of squares change plots. So in here, instead of plotting the within sum of squares, I'm plotting the difference or the improvement that I get from going from one cluster to two clusters. So this distance here is what I'm plotting. From going from 1 to 2, we improve over 0.2. from going to 2 to 3, we improve a little bit less. To 3 to 4, we improve a little bit less, and so on, until there is a point in which adding another cluster is always improving me a similar amount, decreasing a similar amount the within sum of squares. And this is an indication that the improvement that we are having is not due to a better separation of the points, but due to the fact that we're just adding another cluster. So what we want to find is the point just before we reach this flat. So in this case, we would say that for this particular data set that I was using, 6 would be the adequate number of clusters. Within sum of squares change is just one method to obtain k-means. There are p-value based methods, but none of them is really standard right now. There's not agreement on what is the best method, because there is discussion about, well, topics of whether things should be grouped in definite numbers of groups or things like that. So this is not a statistical test per se, but it will work best in most scenarios, in most cases. But you always need to take it with a pinch of salt. Now, I was explaining k-means and PCA together because PCA is a great way to visualize k-means. With PCA, you can transform very multidimensional data into bidimensional visual representations of that data, and then you can use k-means to group the data in that you have into a certain number of groups, and you can plot those groups, in this case as colors for the points, into these PCA planes. And here I did PCA and k-means both with unscaled data and with scaled data, and you can see that the results are a little bit different actually. With that, we finish non-hierarchical clustering, and we go, non-parametric clustering, and we go to parametric clustering. And we're going to talk about a model that is generally known as structure. So parametric clustering is just a model-based clustering, and instead of just doing mathematical things with the data, we create a model that explains how our data was created. Pritchard et al. in 2001, I think it is, created an admixture model that is more generally known as structure because it's the name of the program that they created. And what it does is assigning probabilities of each sample of belonging to an ancestral population. So, in this case, we asked the program, okay, what is the ancestry of my data if my data comes from 3 ancestral populations? And then it says for the sample 1, it has 50% probability of coming from population 1, 10% of population 2, and so on. And you can prove actually that these probabilities of belonging are equivalent to the ancestry of those samples, so that this is equivalent as saying sample 1 has 50% of the genetic material from population 1, 10% from population 2, and 40% from population 3. And you can represent these matrices, these probability matrices, as bar plots like these ones, or maybe some one that you will be a bit more familiar with if you have read the genetics book that you were given, I think, for some course. I don't know. So in this case, each column is an individual, and the colors represent different ancestries from different parts of the world. And you will see this individual, for instance, is 50-50 Western African and Southern African, whereas this individual on the other side is mostly European, with a little bit of Southern African, Indian ascent, and a little bit of the other ones, of course. So this is another form of clustering, but it can only be performed on genetic data. So for this analysis, we will use genetic data of the Brassica species. Now, actually, structure works in a similar fashion as k-means clustering. You can also provide any number of clusters that you want, and then you need to evaluate to decide which number of clusters is better. To do that, the program we're going to use uses a parameter that calls the entropy criterion, and basically you need to look for the number of ancestral populations with a smaller entropy criterion. What the entropy criterion means is if I repeat the clustering with, let's say, 6 populations, do I always get the same results? Are my results stable? If they are not stable, then I will have very high entropy. If they are very stable, I will have a very low entropy. And you can prove that the closer you are to the true number of ancestral groups, the lower the entropy criterion will be. And I think that's actually it for this part on clustering. So yeah, in this case, we would choose 3 as the true number of ancestral populations. So just to summarize, we can use scaled or unscaled data to create distances. I've only been speaking about distances of samples with samples, but you can also take distances of variables with variables in order to study what is the relationship between the variables, which ones are closer to each other. And you can use those distances to create hierarchical clustering. And then the non-parametric methods, PCA and k-means, can be used with any matrix. So with the data matrices, with the distance matrices, with scale data or unscaled data. It doesn't matter. And in fact, there are, so for instance, using the data matrix with scaled distances and creating the principal component is the same as doing the data matrix directly and scaled. I don't know. I will take this out from the video. And finally, you can use only with genetic data the admixture model structure to obtain another type of clustering that is based on probability. So it's an admixture clustering rather than these clusterings that strongly assign each observation to one group. Finally, I would like to give you a bit of a reflection on the difference between phenotypic and genotypic data. So phenotypic variables tell us something about the physical aspect of a plant, and they speak about the physiology or the adaptation or domestication of that plant. So in the end, when we look at traits, what we're looking actually is at the trait selection that has been going on in these organisms for a longer time. On the other hand, when we look at genetic variables, we're talking about markers or sequences. The information that we're going to get is about the phylogeny or the evolutionary relationships or the domestication history of those organisms, and therefore the information that we can obtain has more to do with the origin and the relationship of organisms with each other. This is very important because in the end, if we cluster based on phenotypic variables or if we cluster based on genetic variables, we will obtain different results. With one, we will see those organisms that are phenotypically similar, and with the other, those that are genetically similar. And there is a very good example in Brassicas, in fact, with Brassica rapa and Brassica oleracea. So a study was done here in Wageningen where they analyzed a lot of varieties of Brassica, and they showed that although genetically they are different, Chinese cabbages and European cabbages, which phenotypically are very similar, they originate from different species. Therefore, if we would cluster all these varieties phenotypically, Chinese cabbages and European cabbages would end up together, but if we cluster them genetically, they end up separate. And yeah, this is, I think, an interesting reflection because you will probably see similar things in the data that you have today. And with this, we are done with statistical explanation, and I will see you in the short video where I will explain you how to get on with the practical.